Hello there, Turrentine Science students. This is Mrs. Geis, and today we're going to be exploring biotechnology, also known as biotech. It's an exciting field, and I think you're going to enjoy this unit. Go ahead and take out your green Biotechnology LearnEd notebook and open up to page 6. We'll begin our guided notes together in a moment. Remember, it's important to familiarize yourself with the vocabulary that is on the inside front cover of your notebook, as well as the flashcards that are found on the back of your notebook. Those are great study tools if you cut them out and store them in a baggie. It makes that an easy way and a fun way to study and learn the material. Remember, your teacher will also be going through the unit organizer, the bell ringers, and the unit review questions together with you during class. All right, open up to page 6 and we'll begin our guided notes. So we'll first start with Intro to DNA. All living organisms have a genetic code that provides the information to express all of their traits. DNA provides the code for all life. Deoxyribonucleic acid is the long word for DNA. We use DNA for short. It is located in the nucleus of eukaryotes. Remember, eukaryotes do have a nucleus. That was an easy way that we remembered eukaryotes versus prokaryotes in our last unit. Pro rhymes with no. Prokaryotes do not have a nucleus. Eukaryotes do. Think of you and do. It is located in the cytoplasm of prokaryotes. It provides the information necessary to produce proteins, which allow for gene expression. This makes traits appear in offspring. The history of DNA is fairly recent. In the early 1900s, scientists discovered DNA is made up of repeating nucleotides. Nucleic acid makes up nucleotides. Each nucleotide contains 3 parts: sugar, known as deoxyribose, a phosphate group, and a nitrogen base. Adenine, thymine, cytosine, and guanine. Sequence of nucleotides determines the traits, and the traits are coded for by genes. A gene is a segment of DNA that codes for a trait. In any cell, adenine goes with thymine and guanine goes with cytosine. We'll talk about that more in just a minute. In 1952, Rosalind Franklin studied DNA using X-ray diffraction. She took a famous photo of DNA, and it's called Photo 51. You are welcome and encouraged to use a search engine to look up Photo 51 and see the interesting discovery that she made. She was able to investigate the shape and structure of DNA. In 1953, Watson and Crick came along and investigated the structure of DNA in further detail. They are credited with discovering the double helix shape. If you've ever driven downtown in Burlington and seen the sculpture outside of the LabCorp headquarters, you know what the double helix shape looks like. Here we go. This is the double helix shape that we refer to. This double helix structure basically has nucleotides linked together to form DNA molecules. Sugar, which is deoxyribose, and phosphate compose the backbone. Nitrogen-base pairs compose the middle. Adenine always pairs with thymine. Guanine always pairs with cytosine. This will make more sense when we get into our chemistry unit in a few months. But for now, just know that adenine is always with thymine. Guanine is always with cytosine. DNA condenses into chromosomes which are in nearly all of our cells. Normal humans have 46 total chromosomes. These give the detailed information about all of our traits. All living things have DNA, even some non-living things, especially a famous virus that has been in the news recently. Take a moment and answer questions 1 through 3 at the bottom of page 7 before turning the page. So you have learned that genes are segments of DNA that code for a trait. Your DNA is found in the nucleus of nearly every cell in your body. Think back. Remember when we looked at cheek cells under the microscope? Those cheek cells had the nuclei clearly visible. Your DNA is found in those cells. The only exception would be red blood cells. Because they lack a nucleus, that allows blood to be donated to others. The DNA is coiled and tightly packed in the nucleus. Just before the cells divide, the DNA must copy itself so that all of your cells have an exact copy. DNA composes every gene that you have. Every gene you have is located on a specific chromosome. Each one codes for an individual trait. In class with your teacher, you will be researching the relationship between DNA, genes, and chromosomes. On page 8, you will provide a drawing and a brief description of each. Genetics involves the study of inheritance patterns. On page 9, during class, your teacher will describe what to place in the 3 green boxes. Each one will be labeled with inheritance at the top, phenotype in the middle, genotype at the bottom. Your teacher will give you more details during class. You can also find these terms on the back of your notebook using your flashcards. Bacteria are living organisms that also contain DNA. How do you think they may reproduce? Can bacterial strains inherit traits? Bacteria have proven to be highly beneficial in biotechnology and many other aspects of life. They have many medical, ecological, industrial, and research-related uses, as you can imagine. Now let's talk about environmental factors. Environmental factors strongly relate to gene expression. Gene expression is the physical appearance of an organism's traits based on the proteins they produce. Organisms may develop beneficial mutations to assist in their survival. Sometimes harmful effects can be caused by the environment. In class, your teacher will explain how to complete the chart on page 10. Let's talk about the field of biotechnology now. On page 11, biotechnology is the application of technology involving living organisms to achieve a beneficial purpose. This involves genetics and living organisms. Simply stated, DNA is manipulated, or new DNA is made from piecing together small fragments from other sources. Recombinant DNA, any DNA that has been broken apart or shuffled. Biotechnology involves recombinant DNA, manipulating DNA in some way, transferring DNA from one organism to another. Vectors. These are mechanisms used to transfer DNA, moves the DNA into new cells. So think of this, think of it this way, or this image. A gene gun can shoot DNA into a new location. That's a good way of thinking about it. Biological vectors are living. They may be used to transfer DNA. The fields listed in the chart on page 11 have been significantly impacted by biotechnology. Brainstorm ways that they may benefit from the latest developments in DNA research. The 4 fields to keep in mind are healthcare, forensics, agriculture, and ecology. These are the 4 main fields impacted by biotechnology. Your teacher will go over what to place in the box at the bottom of page 11. Now let's look at biotechnology tools on page 12. We have cells, genes, and proteins. What are they? How are they used? How do they benefit society? Well, cells, genes, and proteins are essential components of the field of biotechnology. You have learned about what they are, but you must figure out how they apply to biotechnology. Why are they important and how can they benefit society? Use the following information on the following pages to complete the chart on page 12. How are these tools in biotechnology? First, we'll talk about our cells. All living organisms are composed of cells, which are considered the basic unit of structure and function for life. Cells contain DNA, which codes for the traits of all living organisms, from unicellular bacteria to multicellular humans, and everything in between. In biotechnology, cells and their genetic material are manipulated to achieve a purpose. For instance, cells can be reproduced in a lab to regrow tissues. Stem cell research is also a very important part of the growing field of biotechnology because they may be used to treat or cure harmful diseases or disorders. So take a moment and fill out the column for cells on page 12 using this information and any other information you may find. Next, we'll talk about genes in the second column. All living organisms contain DNA, which codes for all of their traits. Genes are simply segments of DNA that code for a specific trait. You have genes for every trait you possess, from hair color or skin color to the shape of your knuckles. Bacteria, plants, fungi, protists, and animals all contain genes to code for their traits. These genes can then be manipulated or transferred from organism to organism. For instance, the insulin-producing gene from humans can be transferred to bacteria in order to manufacture larger amounts of insulin for diabetic patients. So go ahead and use this information to complete the column for genes on page 12. The third column is proteins. Proteins are one of the most important molecules for the function of living organisms. We learned this during our health and disease unit. We consume them in our diets, our cells produce them for gene expression, and they even build our tissues. We are full of proteins. In biotechnology, the final proteins produced are very important. A scientist can manipulate the DNA of certain organisms to produce different proteins than they naturally would have produced. For instance, a farmer can grow crops that resist freezing because of a special protein they can be made to produce. So complete the column for proteins on page 12. Now we're going to investigate another important field of biotechnology, the field of forensics. Forensics on page 13, uses science to investigate crimes or crime scenes. Biotechnology in crime scene investigations includes DNA fingerprinting and analyzing and comparing DNA fragments that are left behind. It's created with gel electrophoresis. This is the separation of DNA pieces through an electrically charged field. The restriction of enzymes. We know that enzymes break things down. It's used to cut DNA into pieces. Smaller fragments travel farther into the gel. Each person's DNA has the same banding pattern. So here's an example. The following is a DNA fingerprint produced by gel electrophoresis. When we say fingerprint, we don't mean prints off of your finger, but rather a pattern that is taken from the DNA left at the crime scene. So who is the culprit according to the gel below? Here is the crime scene DNA. This is what was found. Now look at this pattern and see if you see the same pattern in suspect 1, suspect 2, suspect 3, suspect 4, or suspect 5. If you guessed suspect 3, you were correct. It has the same pattern, the same DNA fingerprint, we call it. It may be also used for paternity testing. That means identifying similarities in the DNA of parents and offspring. May also be used to catalog endangered species. The benefits. This helps organize information, discover unknown information, and identify species. Now we're going to talk about another very important aspect of biotechnology, and that is healthcare. So if you turn your notebook to page 14, we'll talk about the field of healthcare, which is, again, very much in the news right now with the coronavirus pandemic. So healthcare, this is the application of biotechnology. It is spreading the most rapidly. It uses genetic engineering, using DNA technology to modify the genetic makeup of an individual. This leads to advancements in healthcare. The Human Genome Project, which we mentioned very briefly during our previous unit. This is a 13-year-long project involving many scientists. Scientists were able to sequence the 30,000 genes found in the human genetic code. The human genome sequencing means mapping every single gene in the human genome. Sequenced pairs of DNA. This allows knowledge of the detailed information in DNA. It is now possible to pinpoint the exact locus of genes that cause diseases or disorders. Just imagine how might this benefit the healthcare field. Gene therapy. This involves replacing malfunctioning genes with functional genes. This enables treating of specific genetic disorders or replacing defective genes with functioning genes. What type of disorders do you think can be treated with this type of technology? Severe combined immunodeficiency, or SCIDS for short. In this illness, patients lack a portion of a gene that controls their immune system. Scientists can attempt to replace that missing sequence with functional DNA. Do you think this could present any challenges? Cystic fibrosis is another fatal disease where a mutated gene causes excess mucus production in the lungs, leading to severe complications and often early death. Scientists are attempting to treat it with gene therapy. Do you think this could present any challenges? One of the drawbacks it's a difficult process. It cannot be done in every cell. Transgenic engineering. This is transferring DNA from one organism to another. So an example would be producing human insulin from bacteria. How it works. Well, the gene for insulin production is inserted into a bacterial cell. This is known as a DNA plasmid. Bacteria can then produce human insulin. Imagine how life-changing this is for diabetics. Other medical uses. Cancer. Being able to predict the risk of cancer in an individual, diagnosing cancer, and treating cancer caused by specific genes. Heart disease, diagnosing, finding genetic links, and using gene therapy to treat heart failure, and stem cell research. AIDS, treating infected cells, using gene therapy, antivirals, and identifying infected cells. Genetically engineered organisms, also known as genetically modified or GMOs. The purposes are often in agricultural contexts and pharmaceuticals. An example would be transgenic organisms producing human insulin from bacteria. Agricultural applications, genetically modified crops. The purpose of this is to increase productivity. The benefit would be allowing for longer growing seasons and a better yield, a better outcome in the number of crops present. Other examples, discuss with your classmates some other ways you think this could benefit the agriculture and food science industry. We will be having debates in class on issues in biotechnology. Conservation, using a variety of methods to preserve and protect natural resources, such as bioremediation. This is the process of removing pollution from contaminated environments and water. It's done by introducing natural organisms. In class, your teacher will discuss the activity on page 16. While we are in a virtual setting, we will not be doing the lab on enzyme actions, unfortunately, but we will try to simulate some labs so that you can see DNA in strawberries and also your own saliva. Let's turn now to Biotechnology Careers on page 18. Advancements in the field of biotechnology are opening doors for career opportunities. Traditional specialties within biotechnology include medicinal, that means improving healthcare. Examples would be production of pharmaceuticals, gene therapy, diagnosis. Agricultural, improving crop yield. Some examples would be GMOs for pesticide resistance and frost resistance. And industrial, creating products for market. Example, cheese, bread, livestock feed. Environmental, impacting conservation efforts. Examples would be bioremediation and the development of alternative fuels. Research-based careers in a rapidly growing field that will include scientific research is crucial for progress. They are needed for understanding cellular components and for solving current issues. You have already learned the basics of biotechnology and biotechnology careers. In the area below, give an example of each. Be sure to review the brainstorm chart on page 11. How have your opinions changed? You can record them on the bottom of page 19. I'm sorry, page 18. Bioremediation is solving environmental issues with the use of biotechnology. It does not involve changing an organism's DNA. The historical discovery of bioremediation was made with the observation of oil spills, indicated that oil was capable of clearing on its own. Naturally occurring bacteria were discovered to be using the oil. They were thriving off the nitrates in the oil. This helps with oil spills. The bacteria are added to the ecosystem if an oil spill is present. Researchers have been developing ways to use naturally occurring organisms to help with excess pollution in ecosystems. Bioremediation in air and water, using bacteria to eliminate harmful toxins. Nutrients must be supplied for the bacteria to survive. Different types of living organisms can be used in different situations. Look at the prompt at the bottom of page 19. Go ahead and respond with what you think based on what you know. Finally, ethics in biotechnology, and we will be having debates in class on this topic. Ethics refers to the moral principles that influence a person's behavior. Biotechnology is very beneficial to society, but it can also present many ethical challenges. Using the diagram on page 20, in class, your teacher will explain how to organize your thoughts on biotechnology related to the issues in ethics today. You will be able to use specific examples from this unit. So that's it. You will be instructed to complete the unit review on pages 21 and 22. And there is also a space on 23 through 25 for notes, as this is an exciting field, especially in the year 2021. I appreciate your listening. I look forward to seeing you in class soon. Have a great day.